will continue our discussions the clinical chemistry, the rest of the last lecture. The final topic that we were discussing it is the horseradish peroxidase, and we know that the horseradish peroxidase actually it's not referred to the horse or radish. Actually it's referred to the mare radish, which is a plant. Its enzyme that is act as a peroxidase and on the heme protoferrin calcium enzyme. Okay, what's so important about it that is used in ELISA, it used also in the Western blot, it used in the immunohistochemistry, so it used on all these stuff. Oxidize substrates using hydrogen peroxidase, so producing color and light. It has, this is enzyme horseradish peroxidase, it has multiple isoenzymes, but the most common is the type C of horseradish. It's a large alpha helical glycoprotein which binds heme is as a redox factor. Um, so it turns the chromogenic substrate, which is the second antibody in our experiment, and turn it to the colored product or light. So we can use the fluorescence or we can use the horseradish peroxidase, and then that is produce maybe the light or produce also the color. The fluorescent dye, it's not common. So we were talking about the Western blot that is composed of the many steps. The first step is the electrophoresis for separating of the proteins, and then the second step is the detections of these blotted membranes once they are on the membrane. I said also an important information it's important note about the methanol that is used as a transfer buffer, which is so important to to transfer these proteins onto the membrane, that facilitating the binding of this protein in this membrane, which is can maybe BVDF, which is polyvinylidene difluoride, or nitrocellulose. This step that we are discussing it is that we have here the membrane after the transfer, and then we make any blocking, and we said that the blocking we can use it uh, like we use the not free fat milk, that's mean the milk without the fat, or we can use BSV for blocking. What is the BSV that we discussed it before? BSA, sorry, not V, is called bovine serum albumin. It's actually about the albumin, so we can say it's a bovine serum albumin. So the bovine serum albumin, or a free fat milk, we use it for blocking. What I mean exactly by blocking is preventing unspecific binding. Okay, so blocking by the free fat milk, and we do the blocking also by the bovine serum albumin. If you just because I really forget what is a bovine serum albumin, we just remember that it's a serum albumin. Okay, then we do specific antibody, this binding to the interested protein, and then we make or we give also the second antibody, which is labeled with the horseradish peroxidase, and we know what is the horseradish peroxidase, and then it produce the light. That is was our topic. Then I talk about the another topic which is so important also. Here we have D Schleicher aldehyde 3 phosphate. Okay, why we say D Schleicher aldehyde 3 phosphate? Because OH on the right side, and we have also the phosphate group that is present on the third carbon, so we can say it's a 3 phosphate. So D Schleicher aldehyde 3 phosphate by the enzyme is called Schleicher aldehyde 3 phosphate dehydrogenase. When I said dehydrogenase, so what is the calcification? Calcification is one under the oxidase reductase enzyme. Okay, then this enzyme, which is known as a Schleicher aldehyde 3 phosphate Schleicher aldehyde like this, and then 3 phosphate dehydrogenase enzyme uses NAD positive plus phosphate group and turn it to the NAD H plus H positive, okay, and then it will turn it to the D13 diphosphate glycerate, okay, diphosphate glycerate. So by this enzyme that catalyzes the phosphorylation of a Schleicher aldehyde during the glycolysis, okay. So again we have here D-Schleicher aldehyde 3-phosphate by the enzyme is called Schleicher aldehyde 3-phosphate dehydrogenase that uses NAD positive plus P positive to produce NAD H plus H positive, and then the results it will be D13 biphosphate glycerate. But the question is, which is so important for the glycolysis, that is causes the phosphorylation of a Schleicher-aldehyde. As you can see, we add a phosphatic group instead of 1 phosphate, it will be 2 phosphatic group. That's mean more covalent bonds. That's why we said D13 biphosphate glycerate. Okay, but so special about this enzyme that is housekeeping gene. It's one of the housekeeping enzyme, and I want just to describe what is the housekeeping gene. The housekeeping gene, they are obtuously expressed. What that's mean, or what that's mean word, that's mean it will be found everywhere. That's what we can say housekeeping, because it should be present everywhere. The gene, which is a sequence of a nucleotide DNA that is code for the protein, because we know that the most of the enzymes are a protein. The genes that is always expressed and always present everywhere is called housekeeping gene. But there are some genes that they are not expressed. They are present, but they are not expressed. It's called oncogene, that this causes the cancer, for example. Okay, but even if they are expressed, then that it will turn to the cancer. So housekeeping gene, these genes that they are always expressed, so we call them housekeeping gene. But some genes that they are not expressed in the normal, which they are called, for example, oncogene. Gene expression results in the formation of the polypeptide by joining amino acids. Gene expression lead to the formation of the proteins. Proteins is about many amino acids that they are bound together with the peptide bonds. And some genes which they are always expressed as their products constantly need for the cellular activity are called housekeeping gene. Again, what is the housekeeping gene? The gene that is always expressed. Okay, so housekeeping gene is the gene that is always expressed, and it's an important for the cellular activity, and the best example for that is our enzyme here, which is called D-Schleicher aldehyde 3-phosphate dehydrogenase enzyme. Okay, other thing, like when I said they are constitutively expressed, that does mean they are always turned on. They are expressed all the time without needing to be told. And the common examples of that, like Schleicher aldehyde 3-phosphate dehydrogenase enzyme, and we have also actin, and we have also tupelin. Okay, so the Schleicher aldehyde 3-phosphate plus NAD positive plus phosphate, by the action of Schleicher aldehyde 3-phosphate dehydrogenase enzyme, it will turn to the D13 diphosphate glycerate plus NADH plus H positive. Okay, and other information about these things, that Schleicher aldehyde dehydrogenase is the key regularity enzyme, important role for the glycolysis and metabolism. It considers as a housekeeping gene. It's actually the sixth step of the glycolysis. Worked in a glycolysis, that's mean generate ATP, and works also in the metabolism, like regulate the oxidative stress, regulate also the DNA repair, regulate also the apoptosis. Okay, and also because it's housekeeping gene, so it's useful to use it as a reference gene. If it's present in the cytoplasm, then it's an important for the glycolysis, but if it's present in the other parts of the cells, it's for non-metabolic reactions. That's actually all the information that we can talking about the housekeeping gene, but we just we repeated every time that the housekeeping gene is the gene that is always expressed for the formation of the protein that is an important for the cellular activity. That's why we called it as a housekeeping gene, and we said the word that is continuously expressed, and it is expressed without even being told. And we said that there are many examples of that, like our enzyme here, the Schleicher aldehyde phosphate dehydrogenase enzyme, or better we can say it's the Schleicher aldehyde 3 phosphate dehydrogenase enzyme is one of them. We have actin, we have tupulin, we have another examples also. What's so special about the Schleicher aldehyde, again to this reaction, that is an important in the sixth step of glycolysis, and you know that the glycolysis is so important for the generation of ATP. As you can see here, the production, it will be D13 biphosphate glycerate, in addition to the NADH and H positive, which they are important for the production of ATP, especially of the Schleicher aldehyde phosphate dehydrogenase is present in the cytoplasm of the cell. If it present in other parts of the cell, then that is used in non-metabolic reaction. So we can say that the Schleicher aldehyde phosphate dehydrogenase enzyme that is not always used for the glycolysis, but it's always used for other reactions like oxidative reactions, used also for apoptosis, used for the DNA repair. So it used actually for other reactions, so it's important. And here it's encoded by the housekeeping gene. It has the same name, which is known as Schleicher aldehyde 3 phosphate dehydrogenase enzyme. Why? Because it is present in many cell or many types of the cell, and is so important it is standard as a reference in the other experiments to compare the other gene levels. It's more active in the cancer and in the neurodegenerative diseases, so it's more active in the neurodegenerative diseases. So remember, that is encoded by the housekeeping gene, which is known as GAPDH, which is known as Schleicher aldehyde phosphate dehydrogenase enzyme, because it is present in many types of the cell, because it expressed continuously without even being told, and it's used as the reference. And here the enzyme that is used for the glycolysis, actually it's important in the second step of the glycolysis. It used also for the oxidative reaction, for DNA repair, for the apoptosis, so it used as a reference gene. It's more active in the cancer and also in the neurodegenerative diseases. And here we have external notes for the SDS page. Actually, we tell, or we have said many information of the SDS page, actually many information about this. So we have here the stacking gel, and we have here the separating gel. Here it's cathode, here is anode, and we wanted the protein to separate it like this. We said that this pH here is 6.8. Why it's 6.8? Because we wanted the proteins to be in one band, and then the pH is 8.8. That's mean we wanted these proteins to be separated. And we have said also that the stacking gel that is composed of the polyacrylamide, that's why we said polyacrylamide gel electrophoresis. So polyacrylamide here is 5%, here is a 12%. Why? Because we said here it will be pick pores, while here it will be small pores, because we wanted them to be separated. And as you can see, yesterday we said that there is glycine. The glycine is the first one, the protein SDS, and then the chloride. And we said that the SDS, that is composed of the sodium, oxygen, and sulfur group, and then the free fatty acid. So it's composed of hydrophilic and hydrophobic, and it's important that it causes denaturations of this protein, that's mean unfolding, and makes all the protein contains the negative charge. And we said we use also beta mercaptoethanol, and we said that is useful for the breaking the disulfide bonds in the bond, so it's important for unfolding this protein. And as we can see here in the stacking gel, actually the protein with the SDS, they are sandwiched between the glycine and the chloride. As we can see here, look how the glycine worked in the acidic pH. I can see the chloride, it's the more negative charge, so it will be the first to go to the anode. So again, in the stacking gel, the protein SD is sandwiched between the glycine and the chloride. Then the pH will be changed, and then the chloride, it's the first always, but the glycine then works negatively, and the protein SDS, it will be the last one. So it's separating in the protein, in the molecular, they are separating depending on the molecular weight, not shape, not the charge, because we said that the SDS make it's similar in the shape and similar in the charge, like this unfolding and the negative charge. So it discontinuous, we can say it's a gel is discontinuous, that form bound to form. The larger protein will stick clearer, that's why we said here it's larger than the small one. In the stacking gel, the protein SDS complex is stacked, so that's why we called it a stacking gel, because it is stacking there. And also we use in the Western blot, actually we use two types of the antibodies. We said that the Western blot started with the gel electrophoresis, and we said that there is here cathode, anode, filter paper, and here the cathode, then here is the gel and the membrane. We said like this is the structure of the Western blot, and then we like explain what is happening, blocking first antibody, second antibody, and this stuff. Actually, we use two types of the antibodies. We use monoclonal antibodies, and we use also polyclonal antibodies, but I will explain in the next lectures more details about them. But just one information, when I use monoclonal antibodies, like a specific one, I'm not looking for the mutation. Okay, I'm not looking for the mutation, because if I wanted to know which gene has a mutation, so I will have several types of the antibodies. I mean, I want to use many types of the antibodies to detect which gene is mutated. So the monoclonal antibodies is not good for using it in the case of detection of the mutation. The better is a polyclonal antibodies. So the polyclonal antibodies, we use it for the mutation detection, and we can't use the monoclonal antibodies for the mutation detection. I will explain then in the next lecture what is the difference between the monoclonal antibodies and the polyclonal antibodies. And then I talked about the blocking in the Western blot. Actually, in the blocking, we used non-fat dry milk, and we use also bovine serum albumin, we use also casein, we use also commercial blocking solution. So we use actually many types of the blocking, but just try to remember non-fat or a free fat milk, and we use also bovine serum albumin, but you can say a casein or something is synthetic, is commercial blocking solution. So about the bovine serum albumin, when the antibodies react to the protein, same, so everything it will be the same action it used for the blocking. What that's mean exactly by the blocking, it means actually preventing unspecific binding of the antibodies to the protein. Okay, and then why don't we use just labeled antibodies instead of using two types of the antibodies? Why I have to use first antibody and then I use the second antibody which is labeled? Because the cascade of the antibodies increased the sensitivity, and if the single antibodies fails to detect the protein, then we lose the whole experiment. That's an important why we use two types of the antibodies, like to make sure that we detect the right protein and we don't miss any other proteins. Or we can explain it because we use two types of the antibodies. Actually, we can use one type of the antibodies. We can use just the label antibody, but actually it's less sensitivity. So we use two types of the antibodies to make it more sensitivity, okay, more sure. So the best, I can say, the best expression of this operation, we can say cascade of the antibodies, like we use type 1 antibodies and then the second type of the antibodies, that is increase the sensitivity of these antibodies. So if the single antibody fails to detect the protein, then the whole experiment is wrong and we lose the whole experiment. So again, it's a good questions and we can ask it. So the cascade of the antibodies increase the sensitivity. The other thing, if the single antibody fail to detect the protein, then we lose all experiment. Okay, and then the other questions is, what is the importance of the membrane beside the gel? Why we have the gel and then we have the membrane? It's helped to protein transfer to the soiled surface, which is a membrane. Proteins are more stable on the membrane and we detect it better, even the small amount. This is other questions that maybe comes to your mind. That's why we use the membrane beside the gel. Why we can use the gel, only the gel, because actually we need a soil surface to detect the protein, and it's important for the protein transferred to be, it's important to transfer this protein to the soil surface, I mean here the membrane. The membrane is so useful for detecting even the small amount of the proteins, and the proteins are more stable in the membrane. And what is facilitating the binding of these proteins on the membrane? Again, methanol. So we use the methanol as a transfer buffer that is facilitating the binding of these proteins on the membranes. Why we use actually the membrane? Because this protein, it had to be transferred into the soil surface like a membrane. It also more stable on the membrane, and the membrane can detect even the small amount of the proteins. And so we can prove the same membrane with the different antibodies. Yeah, we can use, I mean, the same membrane with the different antibodies. What is the denaturation in the Western blot? This is another question. How we can use the denaturation? So the first thing that we heat the sample to the 95 Celsius. So we make it like a boiling, I can say, and then we add in the denaturation agent. So we can say anionic agent, which is known as a sodium dodecyl sulfide, and then it's bind to the protein, and then it's give negative charge, as which is proportional to its length. More large, more negative charge, and we explain that is in the details. The first one, that we healing the sample to the 95%, and then we add the SDS, which is known as a sodium dodecyl sulfate, which is causes unfolding of this protein and also give its negative charge. So the proteins will be same in the charge, same in the charge, and what else we can say, I mean the unfold in the shape, but they are different in the size, or we can say molecular weight, so we can separate it depending on the molecular weight. Then the other questions, why the Western blot is qualitative, not the quantitative? Because qualitative, it means it can detect the protein, but the quantitative, it's mean detect the quantity. So do you think by these questions, okay, by these questions, who is quantitative? Yes, gel electrophoresis, because the gel electrophoresis, I know these are proteins, but I want to separate them depending on the molecular side. I want to know what their molecular side, what is small, what is large, like this. But in the Western blot, it's important for detection of the proteins. It's important for knowing also the molecular size of the protein. So it's many things. It's actually the Western blot is not used actually for only the uh, separation of the protein. It's used for the protein extraction, separation, and detective. That's why we said that the Western blot is a qualitative and not a quantitative. Qualitative because it depends on the band intensity to detect the protein, but it can also tell us, but it can't tell us exactly what the molecular weight. What or who telling us the molecular weight is the gel electrophoresis, like show us what is the molecular weight of it. um, so I think all of these questions are repetitive, but there's, I think these two questions are, I think it's important why we use why we can't use just one antibody instead of two, because the cascade of the antibodies increase the sensitivity, and if the single antibody didn't detect the protein, then the whole experiment, we have lost all the experiment. That's an important. And also why we use the membrane beside the shell, because the proteins, it need to be transferred to the soil servers, and the protein is more stable on this membrane, and also the membrane can detect also even the small amount of these proteins. So I think I explained everything about the gel electrophoresis and also the Western blot, and I think it's enough for them. Um, and I will be in the next lectures, I will explain more about the antibodies. I don't know which lecture, but I will explain more about antibodies. Hope you understand anything. Thank you so much for listening.